Hello, in this video we will see what is action potential. In our previous videos of neuroscience, we have seen central nervous system, the brain and cerebrospinal fluid, and also the structure and functions of a neuron. Here in this we will see what is action potential. Action potential is also called a nerve impulse. It is an electrical charge that travels along the membrane of a neuron, and it will be generated when neuron's membrane potential is changed by signals from a nearby cell. A neuron that emits an action potential or nerve impulse is often said to fire. That means when this action potential happens, we say that neuron fires. And we know the major parts of the neuron: dendrites, cell body, and axon. They play very important role in generating action potential. Dendrites of the neuron receives the information, whereas cell body passes and interprets that information, and axon carry the information to the axon terminal. That is, the signal is transmitted along the entire length of the axon in a phenomena called action potential. So here you are saying the two neurons, one is presynaptic neuron and the another one is postsynaptic neuron. So presynaptic neuron or the sending neuron receives the information from another neuron. Here the dendrites play very important role in receiving the information, and then that information is going to be processed in the cell body of the neuron, and then it passes through axon hillock to the axon, and it is transmitted along the entire length of the axon and reaches the axon terminal. And this, the connection between the two neurons is synapse, or the junction between two neurons is synapse. In that way, the information is carried from the presynaptic neuron to the postsynaptic neuron. So, action potential is defined as a sudden, fast, transitory, and propagating change of the resting membrane potential. Only neurons and muscle cells are capable of generating action potential. As we know, nerves can be very long, often need to transfer or carry the information to very long distances, and it transfers this information to another neuron or another gland cell or muscle cell. That means the information from one neuron is going to be passed to the target cell. That target cell may be a neuron or gland cell or muscle cell. Based on the strength of the incoming signals or stimulation, the neuron decides whether that it should pass through along its axon or not. That is, if the stimulus is strong enough, the signal is transmitted along the entire length of the axon, and that phenomena is action potential. When this action potential happens, we say that the neuron fires. And here the transmission of neuronal signal entirely depends on the movement of ions or charged particles. And what are those ions or charged particles that are responsible for the transmission of neuronal signal? And these ions include sodium, potassium, chloride, and these ions are unequally distributed between inside and outside of the cell. The presence and movement of these ions is not only important in neurons fire but also at neurons rest. So that is, when the neuron is even at rest, it is going to have some potential that is called resting membrane potential. So neuron is at resting state, that means the concentration of sodium ions is higher outside the neuron than inside. So in a resting state of the neuron, this is the neuron. So here outside of the neuron more sodium ions are there compared to the potassium ions, and the relative concentration of potassium ions inside are more compared to the sodium ions. This ionic separation occurs at the cell membrane and creates a chemical gradient across the membrane, and more positive charged ions outside the cell than inside. So this is called electrical gradient, and together this electrical gradient and chemical gradient are known as electrochemical gradient. And now we will see what is membrane potential. So the difference in total charge inside and outside is called membrane potential. So the difference in total charge inside and outside is called membrane potential. So at rest, when no signals are being transmitted, the neuron membrane that is at resting potential is approximately minus 70 millivolts. That means inside of the cell is approximately minus 70 millivolts. That is less positive than outside. That is more positive outside of the cell and less positive inside of the cell. So both chemical and electrical gradients contribute to establishing membrane potential. Inside of the cell has negative charge and outside of the cell has positive charge. So the resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means the concentration gradient and electrical gradient for each ion is equal and opposite. And ions can't simply move across the membrane. Instead, they need a protein embedded in the membrane to facilitate that movement. And most ions cross the membrane through ion channels, and they move through passive diffusion along with the concentration gradient. And there are different types of ion channels are there, like voltage gated ion channels and ligand gated ion channels, and then mechanically gated ion channels. Again, in ligand gated ion channels, extracellular ligands and intracellular ligands. So now we will say what is meant by voltage gated ion channels. Some ion channels are always open in voltage gated ion channels, and many require signals to tell them whether to open or close. For example, voltage gated ion channels, they open only when membrane potential reaches certain potential. So when membrane potential reaches certain potential value, then only they open or then only they close. On the other hand, ligand gated channels are triggered to open when they are bound by a special molecule. So they require some special molecule to open or close. So likewise, mechanically gated ion channels, they open in response to physical forces like changes in length or changes in pressure are going to open these channels. So most of the ion channels are selectively permeable. That means they only allow one or a small subset of ions to pass through. And voltage gated ion channels, for example, typically they allow only a single ion to cross the membrane when they open. So that means that we need separate channels for each ion. That is voltage gated sodium channels as well as voltage gated potassium channels. As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. This is obvious. and if the resulting change in the membrane potential is small, that is called graded potential. And graded potential, they can vary in size and they can be either positive or negative and are transient and typically do not result from the opening of voltage gated ion channels. And when ion channels open and a graded potential occurs, the movement of ions across the membrane creates a local change in membrane potential. And this is accomplished primarily by the use of the sodium potassium pump. And this sodium potassium pump, we know that it uses the energy generated by the ATP hydrolysis to actively transport ions across the membrane against the concentration gradient. In other words, sodium is transported to outside of the cell where its concentration is higher. So here you can clearly see the sodium ions are moving outside of the cell where its concentration is higher, and potassium ions is transported back into the cell where its concentration is higher. So this is potassium ions and this is sodium. And so one cycle of pump transports three sodium ions outside of the cell and brings two potassium ions inside of the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. That is, maintaining this ionic balance in neurons is very important, and this process can account for 20 to 40% of the brain's total energy use. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from minus 70 millivolts to threshold voltage of minus 55 millivolts or higher, and that triggers an action potential at the axon hillock, which then travels down the axon. So this is resting potential, and it is when it goes up to minus 55 millivolts threshold, then that triggers an action potential at the axon hillock, and that action potential travels down the axon. So voltage gated sodium channels, they have three states. One is open, then second one is closed, and third one is inactivated. So at rest, sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position and sodium reaches into the cell because of the electrochemical gradient. As the positive sodium ions enter the cell, the membrane potential becomes less negative and more positive. As it approaches 0 millivolts, this is called depolarization. The movement of the cell's membrane potential to a more positive value is referred to as depolarization. During depolarization, the inner membrane becomes less positive and outer membrane becomes more positive. Eventually, the voltage gradient goes to 0 and beyond 0 up to positive plus 30 millivolts. That is, it is called overshoot. So here you can clearly see this getting stimulus, and then sodium channels open, sodium begins to enter the cell, then potassium channels open, potassium begins to leave the cell, then sodium ions in and sodium channels become refractory, no more sodium enters the cell, and third one is action potential, And fourth one is sodium channels becomes refractory, no more sodium enters the cell, and potassium channels close, no channels reset. And sixth one is hyperpolarization, extra potassium outside diffuse away. So, as the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. It stops the flow of sodium ions into the cell. The change in membrane potential also opens the voltage gated potassium channels, though the open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. The change in membrane potential from a positive to a negative value is referred to as repolarization because the potassium channels are little slow to close for a brief period. The membrane potential is hyperpolarized. It's more negative than the resting potential. Depolarization with corresponding contraction of myocardial muscle moves as a wave through the heart. Repolarization is the return of the ions to their previous resting state, which corresponds with the relaxation of the myocardial muscle. So, an action potential does not get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen. What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain and less frequently in response to a gentle breeze. Some axons transmit action potentials faster than others. One variable that increases conduction velocity is the presence of myelin sheath around axons. And this myelin sheath speeds up transmission through a process called saltatory conduction, in which the action potential's signal appears to jump along the part of the axon covered by a sheath.